All right. There we go. Let's let people get into the webinar from the waiting room, and then I will start. I'll give you, well, it's 11 now. I'll give you another minute to get in. How is everybody today, and where are you from? Go ahead and type it in the chat. Anybody? Ah, there we go. Sanibel and Stuart. Oviedo. Let's see where else. And Orlando. Well, let me go ahead and start so that Dr. Kane has enough time to take your questions after. This is the Florida Friendly Landscaping Program's Homeowner Webinar Series, a series that I give every third Tuesday of the month at 11 a.m. Eastern. My name is Jen Marvin. I am the Florida Friendly for Home Landscapes statewide coordinator for the Florida Friendly Landscaping Program. Today we have Dr. Kane speaking with us about the ghost orchid and his involvement in its conservation. Your microphones have been muted. If you have questions, please type them into the chat box, and I will ask Dr. Kane at the end of the presentation for some answers. Please stick around at the end to take the survey, and it really helps me to give you the kind of programming that you like. And also this year we will have a panel and ask the experts, and I will be taking some of the questions and subjects from the notes in the survey. Next month, our next presentation will be on Tuesday, March 19th at 11 a.m. Eastern on the top 16 houseplants by Liz Felter. So let me go ahead and introduce Dr. Kane. For 34 years, Mike Kane was employed as a professor in the Environmental Horticulture Department at UF, where he taught courses on plant micropropagation. His research emphasis was on development of propagation methods for the conservation of native plants, especially Florida orchids. In recognition of his contributions in 2009, he was elected as a fellow of the Society for In Vitro Biology. In 2014, he has received the SIVB, or the Society for In Vitro Biology, Lifetime Achievement Award. And I know he's done a lot more than that. So Dr. Kane, if you will fill us in and then go ahead and start your presentation.
Great. Thank you, Jen, for inviting me to participate. I always like to share what we've learned over the years and certainly get feedback and questions. And I think that's so important for me to learn. And as was stated, I have had a wonderful time as a employee at the University of Florida, and hopefully today I can show you some of that journey that I've taken. I look forward to your questions at the end, and I'll try to keep it so it's not that long, but I want to include things that are really interesting with these plants, and they certainly don't disappoint. Okay, what I want to cover today primarily is to provide an overview of native orchids, their life cycle, their general threats to survival, but that is a very important point I want to make, because we're in a crisis stage globally with respect to native orchids and the threats that have been put upon them. And closer to home, I want to chat a little bit about our own orchids within our state, and then focus in on how they grow, how they propagate, And each species has its own secrets, and has never ceased to amaze me how the orchids have adapted. And I'll focus then in on our attempts, and most of which have been successful. We still have a way to go on the conservation of the rare ghost orchid, Dendrophylax lindenii. And hopefully at the end, you'll participate in this conversation with your questions. Not knowing the background of who is attending, I know some of you know orchids very well. A couple of people I know who are attending, but I want to talk about orchids in general from a global standpoint, native orchids. Orchidaceae, the largest and most diverse family of flowering plants. There are about 35,000 species. If you look at different references, that number will vary somewhat. They have incredible diverse mechanisms of adapting for breeding, for surviving in a wide range, multitudes of habitats. We have worked on special determining the special pollination systems that are found in different orchids, and that's fascinating in itself. I won't go into that here, but we've done quite a bit of work on that. Orchids in general have extremely small seed. It's minute. It's like dust in many cases, and there are limitations to that. But the plants have evolved very, very specifically to overcome that. And that overcoming the small seeds, we'll go into a little bit later. There is a obligate necessity that specific mycorrhizal fungi form associations with the seed, the embryo in the seed, to help it develop, grow, and establish successfully. And without that symbiotic relationship, it's not always symbiotic, but they wouldn't survive in nature. They're found over across every continent except Antarctica. Within the most of the diversity is found in the tropics, but they're found in the subtropics and temperate areas as well. In the United States, there are about 200 native species. And I mentioned about the necessity of mycorrhizal fungi, but there are other relationships that orchids have in their environment that are critical, but also very fragile. So it's often said that orchid populations, orchid species are the canary in the coal mine. When there are disruptions in their habitat, in their environment, that's where you see decreases in populations and other negative impacts that occur. Many of the factors which are now impacting orchids globally include habitat conversion, which involves, it could be agriculture, urban use impacts. Florida has had an incredible history of doing that. If land goes from natural to agricultural use, impacts may involve changes in hydrology, the use of pesticides, a wide range of impacts that could very significantly impact native orchid populations. Florida is known for its exotic plant invasions and also various other pests, whether it be scale insects, you name it. And these impact native orchid populations. Climate change is a big one, and we're facing that now. We see the difference just in Florida that we are seeing now that are having tremendous impacts on our orchid populations here. The impacts of hurricanes, tropical storms are a major influence of the stability of orchid populations. And then, of course, that we all know about is the illegal poaching of our native orchids. And there are many excited and motivated orchid nerds out there who are so motivated to take plants from the wild. And just about a year and a half ago, there was one where a couple of individuals were caught poaching plants in South Florida, and they were caught. Most of the plants that are epiphytes on trees, when you remove them, their survival rate is extremely low if you don't know how to acclimatize them and reattach them. In many cases, you lose the plant in the end. There are different growth habits that orchids have. There are terrestrial orchids, those that are grow from the ground. Some grow far north. Calopogon grows up in Michigan, the one on the top here. It forms an overwintering bulb or tuber, and so it's protected. So those, we have terrestrial orchids in Florida, and Calopogon is certainly one. Epiphytic orchids are on trees or other Certipodium, the ghost orchid that I'll talk about, is an epiphyte. There are aquatic orchids, including Habenaria species, that are either their feet are wet, and they're found in wet, sometimes soil saturated conditions. Another group of orchids are the orchids which are habitating or colonizing rock, hard surfaces, and they're found attached to rocks. Now, I mentioned orchid seed, and one of the primary characteristics of most species of orchids, not all, there's a few that have larger seed, but the seed is dust-like. The seed coat, which is here, is usually 1-2 cell thick. Depending on the orchid species, the seed coat can be almost crystalline in forms, and they have very different, diverse morphologies, the seed coat. But the point is, the embryo is comprised of only a few cells. It's rudimentary. It could never survive on its own. There's not much of any energy stored, carbohydrate like lipids or starch. They're not there. There's some, but not enough to promote the full development of the embryo into a seedling. And they're typically wind dispersed. And that's one reason why there are hundreds of thousands of orchids produced per capsule. As I mentioned before, orchid germination, successful establishment and growth into a seedling requires a symbiotic association between fungus and an orchid embryo. And we've done a lot of work with this with various orchid species, including the ghost orchid. And what happens is the orchid infects the embryo. It's a source of water, organic carbon by breaking down of organic matter. Orchids, the fungi are decomposes, and a source of nitrogen and phosphorus. Supplying these essential elements, nutrients, allows that embryo that we saw here to develop into the first stage, which is a protocomb. And at some point, the protocomb continues to develop into a green chlorophyllous seedling, where it can produce its own food and essentially provide photosynthate to the fungus. Now, what happens is as a fungus develops within the, let me see if I can go back here. Okay, as the fungus develops, it forms these hair-like masses of hyphae, the filaments of the fungus, called pelotons. And many times as the plant, the seedling gets bigger, it actually feeds on those pelotons. So it kind of feeds on the fungus. So the relationship can change. Now, and we're learning more, there are specific fungi found in certain orchids. Some orchids are generalists, and these fungi are too. There's not just a specific species that is found in a fungus that's found in a single orchid species. There's still lots of work being done on that. So it's a dynamic process. Well, at the bottom, there's a seedling on a tree in a mass of moss. And that seedling, obviously the seed became attached or embedded in that moss. And then it became infected with the fungi, which then allowed it to develop further and ultimately into a seedling, which is photosynthetic. So let's talk about native orchids in Florida. There's about 200 orchid species in the US, and Florida has more than half of those, which is incredible. There are 33 species that are epiphytic, almost 80 that are terrestrial, and there's 3, and I mentioned one of those habenaria, that are aquatic. And at least 60% of them now are at risk, threatened, or endangered. This is a list, and you can't see this one, but this is on the website of Florida's listing partial of the status of different native Florida orchids. And you can see that most of them are either endangered, threatened, or considered commercially exploited, where either the seed is collected or the plants are collected in mass from the wild. And that's quite a few. And Florida is very keen on protecting its native orchids. So given the status of these orchids, and we'll just, we can talk about this globally, there's a critical need to reserve, conserve, protect, in our case, Florida native orchid populations. And this pertains to, or can be applied to orchids worldwide. A good example is my last student, PhD student, graduated in the middle of COVID and worked on 4 species of Ecuadorian orchids. And they have over 4,000 different species in that country, 4,700, I think. So how do we accomplish this? How do we come up with a plan, a strategy, a set of procedures to protect our orchids and restore them? So it's amazing how little we know about the life cycles, the ecology, the needs and interactions of orchids with their environment. So we need a better understanding of this relationship or set of relationships between orchids and their environment. We need to then develop conservation methods, and that's what I'm going to be addressing today for the most part, and then implement these based on science to ultimately manage and promote the long-term sustainability of our Florida native orchid populations. We have so much going on now. Climate change is a big one. If you look at the intensity of the storms, the changes in the temperature range, it's just been incredible. You know, how are these species adapting to that? These are critical bits of information we need to find. I came to the university in 1985 in the beginning, and my background was as a botanist. And I worked with native plants. Actually, it was aquatic plants, and did a combination of field and laboratory work. If you're working on native plant restoration, conservation, you can't stay in the lab. You can't stay in a greenhouse. You have to get your feet wet. And the program that I had, and it was called the Plant Restoration, Conservation, and Propagation Biotechnology Program. That's a mouthful, I'll tell you. But over the years, we've trained many students, both undergrads, and they trained me too. I learned so much from them, with a focus on native plants. And as Jen said during her introduction to me, he's worked on other things, and I certainly have, from native wetland plants to coastal plants. Sea oats is for. So our lab has always been concerned about Florida's natural resources, how to develop techniques and strategies to protect those. Early on, I realized we needed to develop strong working relationships, whether it be with the state. We worked with what was called the Division of Forestry, and we developed a very strong partnership with the U.S. Fish and Wildlife at the Florida Panther National Wildlife Refuge in Southwest Florida. And our goal was to conduct research, provide outreach and education to the citizens of Florida and elsewhere on approaches to successfully protect and conserve our native orchids. This was established in 2005, and we worked with another researcher, Dr. Larry Zettler from Illinois College. He had the Orchid Recovery Program there, and he's a world-renowned orchidologist with an emphasis on the mycorrhizal relationships between fungi and native orchids. And I've worked with him very closely over the years. The person we worked with closely for many years was Larry Richardson. He was the wildlife biologist at the Panther Refuge, and that relationship was so productive in terms of working with students, working with me. And that same is true with Dr. Zettler from Illinois College. So we're very productive in what we did, and I'm very proud of that. But most of all, I'm proud of the students who went on to really excel in their careers. The guy on the right there, Dr. Wen Huang from Vietnam, worked on the ghost orchid, did a tremendous amount of work, and he learned a lot, and I learned a lot from him. So the ghost orchid has been very good to us in terms of develop relationships and allowing us to figure out what makes it tick. Why did we pick and work with the Panther Refuge? It's down just east of Naples. It was created in 1989 through the Endangered Species Act, with a primary focus. The land was purchased to protect the Florida panther and its habitat. Well, its habitat includes native orchids. So with that connection, we're able to develop a orchid restoration conservation program on the refuge there, because it contains over 30 native species, and it's just incredible there. It's a perfect outdoor laboratory. 26,000 acres, part of the Big Cypress Basin. And if you've been, I'm sure many of you have been down there, it is a mosaic of cypress strands, domes, hardwood hammocks, wet prairies, pine flatwoods, and it's just an incredible laboratory. So that's been our laboratory for a long time. Species that we've worked with on the refuge through our cooperative agreements, whether it be doing field studies, pollination biology studies, what have you, include that list there. So we've worked very closely with the federal government, and some of these projects, again, were funded from state funds. So what did Mike Kane do and his group of students in terms of approaching and solving the problem that we're facing? And it's not just about solving the problem by producing plants. It's more than that. That's a short-term solution. If you have to understand the relationship of the plants in the environment, whether it be with fungi, with nutrients. So it's a integrated approach that we took. And you'll see that when I show you some examples of what we did, particularly with the ghost orchid. So we want to get a full understanding so we can develop reasonable solutions that work. And the integrated approach is basically integrating different disciplines of science, whether it be on the top, propagation science, that's what we'll cover today, in terms of how do you grow the plants? How do you grow them, develop them, so that they're successful when you attempt to outplant them back into the wild? On the left there is the heading of genetic diversity. Genetic diversity is good, hygienic, because it allows for stability of the populations. You don't want to be doing anything that will impact or lower genetic diversity, making the population not able to respond to changes. And so we're key to that. How do these plants reproduce sexually, allowing for genetic diversity? So we look at what the pollinators are, whether they can self-pollinate or they require pollination from other plants, and we've done many studies on that to elucidate that. What attracts the insects, the pollinators, and we've done that as well. We talked about the fungi. Critical. If you don't have that microbial association, you won't, in nature, you won't get that growth into a seedling and establishment. So that's another factor. Then the distribution. Why is it that these plants are found in discrete populations? How does that affect the status of the population? And then when you bring all of those things together and you reintroduce plants into a, let's say, a declining population, you bring together all of these sub-disciplines or these disciplines that are integrated to come up with a science-based plan. And that's what we've been working on. This can include, looking at propagation, looking at germination ecology in the field, looking at environmental cues. Good example of that is we looked at Calipogon from Florida to Michigan. And we looked were the differences depending on where they were collected. And oh yeah, you'll germinate seed, and within 2 months, the ones from Michigan form a bulb, overwintering structure. You grow fast and go dormant, or you die, you freeze. So we looked at that, the effect of latitude and geographical source, things like with different species, looking at nutrient comparisons. We've done a lot of that. Again, the fungal relationship. Dr. Larry Zettler has isolated specific fungi from many orchid species around the world. And there are differences. And then ultimately, we want to generate seedlings in an ecologically sound way that will survive when outplanted. There are two germination techniques that we use in the lab. One is called asymbiotic seed germination. And that's where we germinate the seed on a culture medium, this gel medium that contains all the mineral salts and a carbohydrate source, energy source, such that you can germinate them into the seedlings without the presence of the mycorrhizal fungi. This was first reported back in the 1920s by Knudsen, and there are a lot of plants that are orchids that are produced this way. They don't contain or germinated with the fungus. And there's some thoughts that without this fungus, the plants won't survive in nature. Well, that's not true. As you'll see, if you germinate them, they form into self-sufficient plants. You may be able to put them out because the need for the fungal so isn't there once the plants are. So that's asymbiotic. Symbiotic is a little bit more difficult. That's where you germinate the seed, and it occurs in nature, as I talked about a few slides back, or in the lab setting. And you co-culture the seed with the fungus that was isolated from the roots of that species. So in particular, the seeds germinate without a fungus, asymbiotic. It's very reliable because everything's that the orchid needs for germination and seed is in the medium. We've done many studies in optimizing the nutrients in the culture media for growth and development. And it's a rich medium. It has carbohydrates, sugars, glucose, all these sugars, mineral salts. And orchidologists who are growing orchids, they add all kinds of things: V8 juice, orange juice, tomato juice. Bananas are very popular. Coconut water, because these are rich and they contain a lot of organics that could promote the growth of the plants. Now, the medium that promotes germination is sterile. It's been prepared. It has a gel which solidifies it, agar. Well, it's very rich, and if you allow fungal, bacterial spores to get in, it will contaminate the culture. So we want to surface sterilize the seed before we put it onto that rich culture medium, or we'll have failure. And the way we do that is that we put the seed, in this case, I'm just showing dry seed, in a dilute bleach solution, Clorox bleach. And it has a little bit of ethanol alcohol in it. And then you shake it in this tube, and you can use different things. We sterilize the seed. And then and it's only for about a minute. And then we replace the bleach with sterile water. And the seed, depending on the species, the seed floats, and then you plate it out on media that promote the germination of the seed. It's very simple. I showed you the example there where it was dry seed. You can use green capsules, sterilize the capsule, cut it open, then remove the seed from that. The only problem is sometimes that you're not sure when the capsule is mature. If it's not mature, the seed may clump. It may not be fully mature. And you won't get seedlings. You'll get other types of growth that forms clones, which may not be good. So depending on the species, the germination of the seed first forms of the embryo, it forms a protocol, number A, letter A, and then it goes through these developmental stages, including the formation of these trichomes, and then the leafy stage. And we have a series of steps, developmental stages, and it may differ. Usually 1 through 5, and it may differ depending on the species. But these continue to grow, and on the right here, we can see the seedlings develop into leafy shoots, and then we'll put them into the greenhouse. So it can be a very prolific way of growing plants. Now, each of these plants is a seedling. It's not a clone. So hopefully, each of those plants is genetically distinct. That's not always the case. There are exceptions, but for the most case, this is a responsible way of performing orchid conservation with respect to producing plants which are genetically diverse. Symbiotic seed germination. The fungi are isolated from the roots, let's say, of the ghost orchid, which they were, and then they're identified. Today we have some really strong molecular technique for IDing fungi, and we can do that. And then these are isolated into pure cultures. We maintain those on the lower. Let's see if I move this without going off. Here's a fungal culture that was isolated from a native orchid species. And if their seed is co-cultured with the fungus, they form those pelotons that I described a little bit early on. And on the photo on my broad, you can see the mycelia, the filaments of the fungi that has infected the protocoms. Now, the system to do this for mycorrhizal fungi, symbiotic, is a little bit different one. First, we maintain the fungi, pure cultures, on potato depressed agar, that's PDA, and it grows fairly quickly. And then we take a stab of the culture, put it on the center in this petri dish. Now, this petri dish contains oatmeal agar without any sugar. It's just crushed oatmeal. And that allows the fungus to digest the oatmeal. And in doing so, it breaks it down into sugars, carbohydrates, and it grows. Not as fast as it would on the potato dextrose agar. And then it infects the seed that is placed on a filter paper strip right here. And then the fungi grow across the medium, and they infect the seed, and then that germinates. And just like in nature, you end up with a seedling which contains the fungus. So it's a very nice way of developing a system to inoculate the seedlings. Here's some examples here, and you can see the first coat. We usually start them in the dark, then put them in the light. And you can see the mycelia there in the upper center photo, and then the pelotons. And then we put them in the light, and they continue to grow. The seedlings continue to grow. Sometimes the fungi overcome the seedling, killing it. All right, so those are the methods of symbiotic and asymbiotic culture. And procedures have to be modified depending on the orchid species you're working with. But let's talk about the ghost orchid. This is the one everybody is so familiar with. If you are very passionate about native orchids, I would say the ghost orchid is the one that stands out. It's one of maybe the rarest orchids in the world. There are about 1,500 reported in Florida. There's been a long-term study done last, I think, 9 or 10 years at the Panther Refuge, and they went from a handful to about 800 known plants or verified plants at the refuge. The species is also found in Cuba, and that's it for now. And there are about 450, 500 there in Cuba, but they grow entirely different. They grow on about 15 different host species, where ours in Florida grow primarily on pineapple and the pop ash. The interesting thing about the ghost orchid, it's one of 3 shootless or leafless, leafless orchids in Florida. There are 3 of them. And what you see here on the lower right is that the roots serve as the photosynthetic organ. That's where photosynthesis occur. The shoot is reduced to a scale-like appendage on the underside. I'll show you some sections of it. And what it does, it grows a little bit and it keeps sending out roots on the sides. And those attach immediately to the bark, and then they grow out. If you know the history of the ghost orchid with respect to the public's awareness, Susan Olean's book, you may have read that, about the called The Orchid Thief, has really given attention to the ghost orchid. And then sometime later there was the movie, which was a terrible movie with Nicolas Cage, adaptation, again brings light or recognition of visibility of the ghost orchid. As you'll see, the leafless habit of the ghost orchid, it poses challenges to propagating it, we thought, in the greenhouse and then outplanting them. Ghost orchid, when you grow them, will stick to anything, almost anything. But when you're outplanting in the wild native populations, you want to know that you can successfully, they'll attach. Because that attachment is critical for water uptake, either from the bark. The fungus, particularly in the early stage, is critical. We, in detail, looked at seed development, germination, asymbiotically, and the ghost orchid certainly has very interesting seedling development. And DAG is days after germination, and this goes up to 80 days, and we did sections. And there is a shoot. As it develops, there's a shoot that's in there. If you look right here, this is a leaf. And there's the shoot tip. There are leaves, there are shoot tips, there are buds, but they're miniaturized, so you really don't see them, but they're there. We looked at symbiotic seed germination. We were interested in, we had actually 3 isolates of fungi from the roots of Florida's ghost orchid. And we co-cultured them symbiotically. And you can see here that they do become infected with their own isolated fungi and they stay viable. They're not decomposed by the fungus, as you can see them here. They're developing. This is the fungus that's still growing in the plate. Then here are the pelotons, which tells us that we had an infection. The interesting thing is that we looked at seed germination, and there have been studied the rate of seed germination. There have been studies that show that orchid seed grown symbiotically tend to show sometimes enhanced germination over time, and also enhanced seedling development, going from a very simple to a complex seedling. This shows actually 2 isolates. This is 379, the fungus, and 394. 379, which is the right here, sorry, did not enhance germination, though it was isolated from ghost. So 394 significantly enhanced germination. So the isolate you obtain from the target orchids, the individual, may not be effective. And this P7 is a symbiotic control where the seedlings have germinated on a organic supplemented media, but it's not anywhere, germinated not anywhere as fast as it is with 379. So we see very important effects and different effects on symbiotic germination. So we figured out how to germinate them, and then we wanted to look at how they grew in the lab symbiotically, asymbiotically, because we could control everything. And we, like I showed you before, we looked at germination. Here's early seedling development, and then we separate them and step them up into larger containers. And I'm going to, and some of you have heard that, I'm going to tell you the secret of flowering ghost orchids in your greenhouse or in a enclosure, an aquarium, you name it. If you have ghost orchid seed, and you can buy it on seedlings, you can buy them on eBay, and I've seen exorbitant prices for a flat. I mean, sometimes it used to be crazy. Many of the hobbyists are frustrated because they can never form get in a flower. Or if they do, it takes a long time. The key is this: you have to leave them in culture and enhance their growth and let them grow for at least 18 months. And that's what we found. If we leave them in culture for 18 months, they get big, they proliferate, and they form flower spikes. And that's what we're looking at here. And I'll show you a photograph of a flower in nature. And these are bracts, because a flowering spike is a modified shoot. This is from in culture. It's forming tons of the, not tons, but many of these inflorescences. They've never flown in culture, never flowered. For some reason, I never could figure. You take them out, they'll flower. But you leave them in culture. That's the secret. Simple as that. They're just taking out of culture And these are much bigger in culture. You let them grow and get larger, and the size does have an impact.
Dr. Kane,
Yeah.
excuse me, you have about 10 minutes until 12.
Oh, okay. I'm going to go fast. Yeah, I
Okay,
see.
and then after 12, we'll ask the questions.
Okay. Sounds
Thanks.
good. So when you leave them in culture too long, and you have too many of them, and they get large, the roots of different seedlings fuse. Because this is a bad thing, because you can't get them apart. Once they fuse together, there's no way. And if you're doing conservation work, you want to work with seedlings, and you got to be able to differentiate those. So this causes a problem. And the other problem is the roots are growing in all different orientations. So when you go to try and mount them on a substrate, a lot of them are sticking in the air, and many of those never attach. So we asked ourselves, could we cut the roots? And some of you are probably getting very nervous now, because we were, because they're important. And where they're stuck together, can you trim those roots off, prune them off, and will they grow new ones? In culture, this is not. And we looked at growing them at 5, half a centimeter and 1 centimeter. And they grow right out. You can't kill this plant. The shoots underneath will produce new roots 100%. Within 2 months, and they're all growing at one plane. So it makes for a plant you can hopefully attach to a substrate. So that's what you get. So yes, you can do that. Now, the problem with this plant, the ghost plant being leafless, the roots are the photosynthetic organ. So how do you attach it on a tree or in the greenhouse on a substrate? You can't cover it. The plant on the right is a pseudo, which has a massive root system and has leafy shoots. So you can easily mount that to a host tree by putting, moss with fabric or rubber mesh around it so the roots can attach. That's easy. But with the ghost orchid, you cannot permit light to the roots, or you'll totally block photosynthesis. So what do we do? How do we do it? So we have to figure out a way so the root tips will be able to grow onto or the host tree bark with minimal impact on its ability to photosynthesize. So we found a solution, and that's Joanne's Fabrics and Crafts. Now, I'm going to say I'm 71 now, and I had never been into Joanne Fabric until this. And there was my grad student, Huang. I mean, running around Joanne's Fabrics, feeling fabric and putting it up to the light. And people were looking at us strange, but I never thought I'd be excited to go into a fabric store. But we thought this might be a solution, and it turned out to be. It's substrate that we use that may work with something that's biodegradable fabric. So we tested it by mounting the different fabrics, and we also compared using pop ash bark substrates with a hole drilled in so you can put the ghost orchid on it with a cable tie. So we developed these storage container experimental chambers or acclimatization chambers with these racks. And then we had the fabrics, burlap, cotton, all kinds of different types. I think there were about 8 or 9. And we harvested the plants, we mounted them like this, and then we put them in our greenhouse under shade. And they were sealed with venting, that we did have vents. So what we found out, and it was nice, but that actually burlap, washed burlap, worked the best. The plants could put the roots under the holes in the mesh and attach. And we used pine or pieces of wood, wrap the burlap around it, and the plants attached. And this was our first method of growing in the greenhouse and then outplanting. So we grew a lot of these, and we went out, and here the burlap is attached with a hot glue gun, and we then had to pull the burlap off the wood, the block of wood, and then we attached the attached plant. We stapled it onto the substrate, and there you can see it's 4 months, and here's one 20 months where the plants are. Some of the roots don't attach, but they continue to grow. And there's one 24 months. So the system works. We had about 90% survival first year. Second year, this study went 2 years total. There's about 72% survival and a few missing. But that system wasn't quite right because it damaged the plant when you tried to remove the burlap from the block of wood. So I had the idea of making burlap frames, 4x4 frames, and we would harvest the plants from the lab, and we would attach them, with those cable ties, and we put many of them on the frame. They grew very quickly in the greenhouse, and we had incredible amount of flowering. One time there was over 300 plants in flower in the greenhouse. Some would set capsules, but there was nothing in the capsule. And that's probably because it couldn't self. These were all from one capsule. Okay, so that's sexual propagation from seed. That's the best method to use for outplanting. Clonal propagation is not. You should never do that. But there are a lot of horticulturists out there, breeders, who would like to produce plants quickly, not for habitat restoration or conservation, but for horticultural reasons. So while we were doing these studies, we saw that ghost orchid proliferates quickly in culture. You grow them out. When they flower, all of the inflorescences here, the spikes, there's a node, there's a bud at each one of those. Some form flowers, others don't. But if you're familiar with propagating orchids clonally, you can take the inflorescence of many orchids, and then you can treat them with kiki paste or paste with different hormones, and the bud will grow out, and you can, from each node, each bud, you can produce a plant, as you can see in the lower right on decay. So if you remember back, I showed you this photograph. This plant, an older plant, was in culture, and it was forming those inflorescences. You can take each one of those nodes and produce another plant, let it grow out. So if you have a clone, let's say you have a plant that produces a red flower. Imagine that, a ghost orchid with a red flower. It'd be worth a little bit of money. People would buy it. But you need to clone it. That's just one way you can clone it. Okay. Last one is, many orchids in culture form what they call protocorm-like bodies. They form these masses of clonal clusters, and you can divide those up into little pieces, and each one of those will form many new plants. You can produce millions of plants very quickly, and the ghost orchid certainly does that. So there are ways to satisfy the horticulturists and those who feel motivated to go out and poach the native plants. Hopefully this will help deter that. And that's true with other species. Okay, last thing real quick. Last June, the Ghost Orchid got headlined at the Chelsea Flower Show. John Park, who is a very influential developer and finance guy from Naples, was the driving force behind having a orchid conservation booth or display. It's the one on the upper, my upper right, with the two people standing there. It was incredible. It was the orchids of Great Britain, Cameroon, and South Florida. And it was the title was "In Search of Florida's Ghost Orchid," and the orchid was provided, was in bud. It was provided from Chicago Botanic Garden, and it was hidden, not really hidden, but on that tree that was brought in. And it was in bud, but didn't flower. Didn't bud, didn't open. The day or 2 days after the show, it was donated to the Royal Botanic Kew, and it flowered, and it was incredible. All over the world, the news that traveled, the people just flooded the garden there to see it in flower. All right, I think that is it. And do they go over? Oh, no, I didn't.
Oh, only 1 minute over.
Wow.
Okay, so let's see. If you have questions, type them in the chat, please. Let's see here. The first one is a comment that you can probably comment back on. She says, as a member of the Central Florida Orchid Society, I am still wondering what specific fungi have been introduced, or I'm sorry, identified to be successful with aiding in orchid seed germination to a seedling. Understanding it's not just a single fungus. We often hear how orchids die from fungus, so we cringe when we hear orchids being infected by a fungus.
Yeah, that's a good. I shouldn't use the word infected. I need to, because every time I say that, I know it. But that's an excellent question, because, what is the relationship between the fungus that's in the plant at the time you isolate the roots from the roots? And we had a paper, Lanon looked at the fungi that were in the roots down at the Wildlife Refuge. And we had put out our ghost orchids, the first set there. And he went in and he wanted to see if they got infected because they weren't pre-treated. That group wasn't. And he found that there's a common one that was found in the native, a single one, because they use molecular markers to differentiate them. There's a big discussion now whether when the orchids germinate, there may be a germination fungus that's specific for the germination stage. And as they get older, there's a lesser need for the fungus to be there because the plant is self-sufficient. And then maybe there are specific fungi later on that may be important for another reason. We're not clear about that yet.
Okay.
But it's good. Some fungi don't need to be there, and they shouldn't be there. There is a fungus, and we published papers where there's host specificity. If you use a fungus from another species, native species, it may or may not promote germination when grown with the plant. There's so much we don't understand. It's incredible.
Okay, along the same lines, I wonder, with the ghost orchid utilizing the fungus for successful seedling germination, if the leafless approach was developed so it has less complications as an adult orchid with fungus. I could be way off on that, but a curious thought for conversation.
Now, there are three, there are leafless orchids around the world. They're not really leaflet. That if you open them up and look, they're leaves. They're like scale-like appendages, and the shoot tip is reduced. The habit, you think about, the plant is very close to the bark. It's just a root, and it's in the crevices, so it allows for water condensation in the morning to be absorbed by the roots, because the vellum, it's like a sponge. It just condensation, it just sucks it up. So the more leaves you have, the more water loss you have, unless they're really specially adapted. Now and I didn't show this, but the ghost orchid is extremely drought tolerant, extremely. We left some cultures in a vessel, and it's still sterile, but it dried out. And I went into the lab and I looked in, there was quite a few plants that were in there, and they looked like overfried French fries. They were brown. I said, they must be dead. But I said, I put some sterile water, distilled water, and a week later, the tips started growing. They turned green. So we did a big study, and it's extremely drought tolerant. It's extremely. And I'm not surprised, though, many people develop these very specialized growing conditions for the ghost orchid, but in the wild, they're pretty tough. That really astonished me.
Well, we have a couple of comments. Thank you. I have been fascinated by the ghost orchid since I read The Orchid Thief. Not as fascinated or obsessed as La Roche. Also, somebody in Venice said, I had two things that helped mine grow. One, mount with new growth down, and two, mount with dental tape. Won't cut into roots. So
that's a good suggestion. Yeah, there's so many ways you can approach it. That's what's exciting. There's probably a better way. We like the burlap, because if you go out in the field, it decomposes within a year and a half, 2 years. And we compared burlap versus cheesecloth, where we staple it so the plants just push against the bark. And that works. There was no difference in survival, but the cheesecloth, it stays wetter in there. It creates a little cave, and then you get some insects growing in there that I'm not sure long term what that would do.
great. Well, thank you, Dr. Kane. It's always a pleasure to talk to you. We had some other comments. Amazing, fascinating. Thank you. I want to thank you for sharing your knowledge of on this lifelong project that you have.
Yeah. Been a journey. I do have that handout if somebody wants. I will send it to you, and if anybody wants a copy, I'll send it as a file, and then you can give it to them.
Sure. Thank you so much, Dr. Kane.
Thank you.
Thank you. Go ahead.
No, I'm done. Thank you.
Thank you, everybody, for joining us. We will see you next month, and please make sure you take this survey. Thank you, Dr. Kane.
Have a nice afternoon.
Thank you. You too. I'll send you some comments and
Sure.
Numbers and survey stuff later.
Great. Thank you.
Thank you. Bye-bye.